Hello, this is VG Shanmugapriya. Today we will be seeing about restriction enzymes. Restriction enzymes are also known as restriction endonucleases or restrictase. These restriction enzymes are coded by the bacterial DNA in order to protect itself from the invasion of the bacteriophages. The bacteria protects its own DNA from the attack of these restriction enzymes by methylating the restriction sites. Restriction enzymes cut the sugar phosphate backbone of both the strands in a double helical DNA. They have wide applications, the important ones being rDNA technology for gene cloning and protein production, genome mapping, and in RFLP analysis. Restriction enzymes recognize particular stretches of sequence on DNA called recognition sites or restriction sites and cut the DNA either at, near, or little far away from the site. Let's now see about the recognition sequence. Most of the recognition sequences are palindromic in nature. Palindromic is when the sequence reads the same both forward and backward. But in DNA, the forward and the backward sequence are found in complementary DNA strands. Hence, they are called as inverted repeat palindrome. You can see here, this is the recognition sequence of EcoRI. Here you can see the palindromic sequence is split in the forward strand and the reverse strand. The same over this region. Also, there are recognition sequences which are non-palindromic in nature, and a recognition sequence can be divided or undivided. About these two, we will be seeing in the coming slides. When restriction enzymes cut DNA, it results either in blunt ends or cohesive or sticky ends. So we can see here when the cut is at the middle of the recognition sites, smooth cut ends will be formed. This you call it as blunt ends. The sticky ends are of two types, 5' overhang and 3' overhang. When the cut is made at the recognition site, based on the ends of DNA which are hanging at the cut region, is called as 5' overhang or 3' overhang. When restriction enzyme digests a DNA, that is cuts a DNA, the number of fragments formed vary depending on the conformation of DNA. With circular DNA such as vector DNA, the number of fragments formed will be equal to the number of cuts. Whereas with linear DNA like eukaryotic DNA, the number of fragments formed will be one extra than the number of cuts. Next, frequency indicates the number of recognition sites for a restriction enzyme on a DNA, and this frequency depends upon the number of bases in the recognition sequence. We have 5 types of restriction enzymes, of which type 2 is mostly used in molecular techniques. We will be seeing about the first 3 types here. Type 1 restriction enzymes consist of protein complexes which are made up of 5 proteins, 1 specificity protein, 2 methyltransferase and 2 restriction endonuclease proteins. It needs 3 cofactors ATP, SAM and Mg2+. And the site of cut is random. It cuts nearly 1000 base pairs away from the recognition site. The recognition sequence of these enzymes are asymmetric and bipartite sequence. They are mostly non-palindromic in nature. Here you can see an example. The recognition sequence consists of 3 specific bases followed by 6 bases which are of any type and then 4 specific bases. This type you call it as bipartite, which is divided here, and they need 2 recognition sequence. You can see here as this it needs 2 recognition sequence in any orientation. Next is type 3 restriction enzymes. These enzymes consist of protein complexes which are made up of 1 methyltransferase protein with specificity domain and 1 restriction endonuclease protein, only 2 proteins. The cofactors are ATP and Mg2+. The other cofactor SAM is not essential, but if it is present, it enhances the activity of this enzyme. The cut site is 20 to 30 base pairs after the recognition site. The recognition sequences are again non-palindromic sequences. We can see an example here. They are non-palindromes. They seem like repeated sequences. These enzymes, also like type 1 restriction enzyme, need 2 recognition sites, but the difference is they should be inversely oriented. You can see the pattern how they should be. Coming to type 2 restriction enzymes which are widely used, there are many subtypes in it. We will first see about type 2P, which is the predominant one and which is commonly used. The enzyme complex generally consists of homodimer proteins. They do not carry out methyltransferase activity and hence contain specificity and endonuclease activity in a single domain. In this type, we also find monomers and homotetramers. It requires only Mg2+ as a cofactor. The site of cut is within the recognition site. The recognition sequence are palindromic in nature and they are 4 to 8 nucleotides in length, and they require only a single recognition site. These are the other subtypes of type 2 restriction enzyme. They differ from each other in one or the other characteristics. Like they differ either in the number of subunits or recognition site pattern, cleavage site, anything like that. Few subtypes are mentioned over here. Let's see in detail about the different recognition sites of type 2 restriction enzymes. The number of specific bases in a recognition sequence is generally taken as the site length. Now you can see here some of them they cut within the recognition sequence, whereas some of them they cut very near to the recognition sequence. The recognition sequence can be undivided like this or they can be divided like this, having any bases in between. Also we can see degeneracy in the recognition site, which is indicated by the different letters over here apart from your AGCT. The other single letter codes, they represent more than one DNA base. Now let's see about some important terms with regard to restriction enzymes. When two restriction enzymes cut the DNA at the same recognition site, we call them as isosisomers. Neosisomers, they recognize the same recognition sequence, but they cut at different regions. You can see the example over here. Another interesting type is isocaudomers. Here, two restriction enzymes can have different recognition sequences, but when they cut, they create identical overhanging termini. So these cut sequences, when they are ligated, most of the time the recognition site of both the restriction enzymes are lost. So here you can see the recognition sequences are different for these two enzymes. The cut ends are similar and they can be ligated like this. Prototype is the term used to indicate the enzyme which is first identified for a particular recognition sequence. The other enzymes which are identified later having the same recognition sequence are called as isosisomers. There are many labs which synthesize and distribute the restriction enzymes. One of them is New England Biolabs. In their website, you can get information about all types of restriction enzymes. To mention, Rebase is a comprehensive database of information about restriction enzymes. Also, Restriction Mapper is an important online tool that finds restriction sites in DNA sequences. It also provides a virtual digest of the sequence. The link to my video about Restriction Mapper is given below. People who are interested in this Restriction Mapper tool can see to that one. So this is in brief about the restriction enzymes. Kindly subscribe to my channel.